Hi, today we are going to learn about nucleic acids. Here we will understand that what are the difference between nucleotides and nucleosides. We'll try to understand the structure of DNA and RNA and their function. Uh, we'll discuss about semi-conservative replication of DNA and translation in the next lecture. So let us start with the introduction to nucleic acid. Nucleic acids are very large molecules, and it consists of a long chain of monomers of nucleotides. So nucleotides are the building blocks of nucleic acid, we can say, and the DNA and RNA that we know are the example of the nucleic acids. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acids. Nucleic acids are the molecules, and these molecules store information for cellular growth and reproduction. So this is the function of the nucleic acid. It stores all the information which is required for the cell's growth and its reproduction. In this slide you will understand that how nucleotides are made. So nucleotides are made up of three basic structure, three components. First phosphate, second base which is nitrogenous base, and third one is a sugar which is basically pentose sugar. So these are the structure. So you can see the sugar, pentose sugar, which has 5 carbon, and at the first carbon of the pentose sugar nitrogenous base are usually attached with an glycosidic bond, and there is a phosphorus, the phosphate group which is attached to the fifth carbon of the sugar by ester bonds. And in the nitrogenous base, basically there are two types of nitrogenous base, purine and pyrimidine. So purine and pyrimidine, you can see the variation, the difference in the ring, how it looks like. Pyrimidine ring usually have 5 atoms in their structure, or 6 atoms in their structure, sorry, and the purine usually have 9 atoms in their structure. So for DNA, the example of purine is A and G, which is A for adenine and G for guanine, and for DNA, the pyrimidine is C means cytosine and T thymine, and for RNA, purine are same, but pyrimidines here we can see instead of thymine there is uracil. These are the structure of the pyrimidine and purines bases, nitrogenous bases. So you can see the structure of cytosine which are present in DNA and RNA, thymine which is present in only DNA, uracil which is present in only RNA, and adenine and guanine both are present in DNA and RNA. Okay, so the pentose sugar here, we see for DNA the pentose sugar is of deoxyribose, and for RNA the pentose sugar is ribose. What is the difference between deoxyribose and ribose? So here we can see the structure. For ribose we can see which is present in RNA. Here at the second carbon there is a OH, but in the case of deoxyribose present in DNA, here second carbon don't have, there is no oxygen, there is only hydrogen in the place of OH. So that is the only minor difference between the ribose sugar and deoxyribose sugar. Okay. So structure of nucleoside. Now how nucleosides are made. So nucleoside is made up of two basic components, sugar plus base, nitrogenous base. Sugar is a what we say pentose sugar, and the base is either purine or pyrimidine base. So we have the example of nucleosides here: adenosine, deoxyadenosine, guanosine, deoxyguanosine. These are the nucleosides. And nucleotides, we can see adenosine 5 monophosphate, deoxyadenosine 5 prime monophosphate, guanosine 5 prime monophosphate, and deoxyguanosine 5 prime monophosphate. So simply we can see the differences that in the structure the nucleosides are having only two basic components, phosphate and sugar, whereas nucleotides contains three components, phosphate, sugar, and nitrogenous base. So these are the structurally made nucleosides and nucleotides containing different nitrogenous base. So base in the case of RNA, it is adenine, so nucleotides become adenosine, and respectively the nucleotides becomes adenosine 5 prime monophosphate, AMP. If the base is guanine, so nucleoside will be guanosine and nucleotide will be guanosine 5 prime monophosphate. Cytosine, so nucleosides will be cytidine and nucleotide called is cytidine 5 prime monophosphate. If the base is uracil, so nucleoside will be uridine and nucleotides made is uridine 5 prime monophosphate. So these are the nucleosides and nucleotides of RNA. Now in the case of DNA, if the base is adenine, so here nucleosides made is deoxyadenosine and nucleotide will be deoxyadenosine 5 prime monophosphate. So these are the what here we are seeing that there is a deoxy prefix is added in the case of DNA nucleosides and nucleotides, because the sugar here is a deoxyribose. So that is only the difference. So different components of nucleotide, we have learned that nucleotides are made up of three basic component, phosphate, sugar, and either of the nitrogenous base, either it can be purine or pyrimidine. This is the primary structure of that nucleic acid that we are going to know, that nucleic acids are made up of polymers of nucleotides. Okay, nucleotides are joined together by phosphodiester bonds. So here there are two nucleotides which are going to join and it forms phosphodiester bond. The link between two nucleotides are called phosphodiester bonds. So 3 prime of 3 prime, the first nucleotide you can see the 3 prime, there is a OH group present on the sugar and this will bind with the phosphate group of the second nucleotide and it will create phosphodiester bond. A nucleic acid has the long chain nucleic acid has a 5 prime phosphate and 3 prime OH group free at their end, and a nucleic acid is read from the free 5 prime end using the letter of the bases, like the basis is A, C, G, T. So accordingly we can name the nucleic acid component. The structure of DNA here, what we see here, that the building blocks are made up of basically sugar and phosphate, and sugar phosphate it makes the building block. In DNA, nucleotides containing bases A, C, G, and T are linked by phosphodiester bonds between deoxyribose sugar and phosphate groups. So nucleotides are linked by phosphodiester bond. This is the primary structure of RNA. Here we can see the nitrogenous base A, C, G, U. We can add C, G, T here. Thymine nucleotides are only present in DNA. The nucleotides in RNA is linked by here also phosphodiester bonds. Okay, and here we see the sugar is ribose. Okay, so this is the two differences that we see in the structure of RNA and the structure of DNA. Sugar and the nitrogenous base is here uracil. Okay, ribose sugar and uracil are present in the case of structure of RNA. Double helical structure of DNA, we can see the two DNA strands means two structure, two polynucleotides, they uh, wind each other and it forms a spiral staircase like structure, and this is a double helical form of the DNA, and both DNA strands are linked together by hydrogen bonds. Okay, the two DNA strands, they are attached to each other through the hydrogen bonding between the bases. So there is a two hydrogen bonds between adenine and thymine, and there are three hydrogen bonds between guanine and cytosine. And the base, the A is always linked to T and G is always linked to C. So we can say the bases of one strand are complementary to the basis of another strand. So this DNA model of replication will study in the next class. Thank you.